So, this auditory lecture is about the introduction to microbiology. So, you're enrolled in a class called microbiology. So, microbiology is the scientific study of microscopic organisms and viruses. So, there's a reason there's and viruses on the end, and that's because viruses actually don't fulfill the characteristics of life. So, microbiology is the scientific study of microscopic organisms. Organisms imply life, and viruses don't fulfill the characteristics of life. For example, they don't reproduce on their own. So, we have to put and viruses on the end of the definition of what microbiology is. Microscopic organisms, here in microbiology, we often call microbes for short. Microscopic organisms and viruses have to be viewed with a microscope. You can't see them with the naked eye, and they are virtually everywhere. The cool thing about microbiology is every time somebody thinks microscopic organisms can't live there, microorganisms seem to always prove us wrong. So when I was going to college, and I'm old, they said that the stomach was sterile. So, if you look back in medical schools and microbiology textbooks, it used to say the human stomach was sterile because it's full of hydrochloric acid, which has a pH of 2 to 3. We all know that because we've all thrown up, and then your throat burns and stuff like that. So they were like, there's nothing that could possibly survive in the human stomach. But it turns out that there is a bacteria called Helicobacter pylori, which actually lives in the human stomach and thrives there, and it is the cause of ulcers in an awful lot of people. And that turns out to actually be weirdly kind of a good thing because ulcers can often be treated for a lot of people with antibiotics because it can be caused by Helicobacter pylori, because it can actually be caused by bacteria. We have found bacteria living in Old Faithful, a bubbling, boiling geyser. Again, you would think there could be no bacteria there because how do enzymes, which are usually made of protein, survive in boiling water? Those bacteria have enzymes that have adapted to survive in that environment. There are bacteria that live on the bottom of the ocean in thermal vents. There are bacteria that live in salt licks and pretty much everywhere that we've ever looked. So there are microorganisms virtually everywhere that we look. Microorganisms get some really bad press. So I think if you asked people, what percentage of microorganisms are pathogenic or disease-causing, I think most people would pick an extremely high number because I think most of us, when we think of microorganisms, we automatically link microorganisms and disease. But that's actually not the case. And really, what microorganisms probably need is a really good PR person. Because the truth of it is that the vast majority, or 97% of microbes that are associated with humans are not pathogenic or are not associated with disease or do not cause disease. It is only 3% of microorganisms that are associated with humans that are actually pathogenic. I mentioned that here in the very beginning. This is a microbiology class that's geared towards people that are going into healthcare. So we're certainly going to focus on those 3% that are the microorganisms that are associated with humans that do cause disease. But on this very beginning conversation about microorganisms, I do want to point out that 97% of the microorganisms that are associated with humans actually do not cause disease or are not pathogenic. So you're enrolled in a microbiology class, which is the scientific study of microscopic organisms and viruses. And now I want to talk about exactly what microscopic organisms actually include, or what microbes include. So it includes fungi, which in microbiology includes yeast, and mold. So yeast, you go to Fry's and you buy a package of baker's yeast and you make pizza dough or donuts or something amazing like that. That's yeast and mold. So we've all gone and bought an amazing package of strawberries, and then later you go to eat them and there's that white fuzzy stuff growing on them. That's mold. Or you go to make a peanut butter and jelly sandwich and you pull out the bread and there's black fuzzy stuff on it or green fuzzy stuff on it. That's mold. Or you have a leak in your shower and there's black fuzzy stuff growing in the shower. That's mold. That's what we're talking about in terms of fungi for microbiology. We're not talking about mushrooms. That's botany's problem, not our problem. Protozoa falls under our microbe umbrella. Protozoa are things like parasites, which we're going to talk about here in microbiology. Then algae. Algae are really important. They live in things like the oceans and lakes and ponds and streams. Bacteria, of course. Helminths, which are microscopic parasitic worms, which we're also going to cover in our conversation about parasites, and viruses. So fungi, protozoa, algae, bacteria, helminths, and viruses is what we include under our conversation about microbes. Here, really early on, I want to talk about normal flora. Some people call it your microbiome or your normal biota. There's all kinds of terms for it. But we actually have about 10 times as many microbes living on and in our bodies as we actually have human cells. There are about 500 to 1,000 different species of microbes living in and on us. That's kind of a big range, 500 to 1,000 different species, but it kind of depends on you and your lifestyle. If you stay in one place, if you grew up just in Arizona or you just stay in the United States your entire life and you never travel outside the United States, you're going to be much closer to that 500 species of microbes. But if you travel a lot more, maybe you finish your degree in nursing and you decide to do work for Doctors Without Borders and you go to some really fascinating and interesting developing countries, you're going to be much closer to that 1,000 different species of microbes living in and on us. The average adult has about 3 pounds of normal flora, and they normally do not cause disease. Some of them, though, are opportunists, meaning if something in you changes, if the ecosystem within your body changes, they will take advantage of that. So, for example, in women, if we go on an antibiotic, we will often suffer and get a yeast infection. That's because we have lactobacillus that are normal flora of the vagina. Going on an antibiotic can kill those lactobacillus bacteria. The lactobacillus bacteria produce lactic acid, which keeps vaginal pH very low. We always have a few yeast in the vagina as part of our normal flora, but they don't like that pH, that the lactobacillus are keeping the pH in the vagina low of, say, about 3 to 4. If those lactobacillus start dying from the antibiotic, then the vaginal pH will rise because they're not producing that lactic acid, and the yeast will actually like the fact that the vaginal pH is raising, because yeast prefer a growth environment with a pH of about, say, 5 to 6, and the yeast will actually start to overgrow, and then women will actually get a yeast infection. Another example of an opportunistic bacteria that we have as part of our normal flora is E. coli. E. coli is normal flora of the intestinal tract. But if E. coli gets access to another part of our body, say, for example, the urinary tract, then it can cause a urinary tract infection because it has access to a new location. Here's some interesting factoids. The human lungs contain about 2,000 microbes per square centimeter. There's about 1 billion viruses in a gram of human feces, and human breast milk contains about 600 species of bacteria, and there's about 100 types of fungi in the human intestine. So, if you're at like a dinner party and the conversation lulls, just, pull out some of your interesting micro class facts and you can get the conversation really going. So the process of acquiring our normal flora actually begins when the water breaks for mom during the actual birthing process, and the vaginal flora can actually enter the uterus, and then we of course again will start to acquire normal flora during the delivery process, and then of course after we're born, from contact with people, contact in the environment, from food and things like that. They've even done some interesting studies now where they're concerned about babies that are born by cesarean section because they don't get access to the vaginal flora. So should they be somehow getting exposed to that bacteria? It used to be that babies that were in the NICU for a very long time, everybody wore gloves and wore tons of PPE, but now there's a little bit of a different thought about that, and parents sometimes are encouraged to actually not wear gloves and to actually have skin-to-skin contact to let that normal flora move across, because we don't want the baby to pick up something like multiple resistant Staphylococcus aureus from the environment. We actually need the baby to pick up that normal flora. Normal flora is really important because it's basically taking up valuable real estate. And if it doesn't take up that valuable real estate, then a potentially pathogenic microorganism could pick up that take up that valuable real estate instead. So life as we know it would not exist without microbes. Microbes produce about 50% of the oxygen that we breathe. Algae produces it, as well as photosynthetic bacteria. So I think a lot of times people think that it's things only like plants and trees and things like that, and we should all go hug a tree for all the oxygen that we breathe, but really we should hug bacteria and algae for it. And actually, it's more than that, because plants actually need the microbes in the soil to help them obtain nitrogen. So without the microbes in the soil, plants couldn't even produce the other 50% of the oxygen that we actually breathe. So it really all comes down to microbes producing the oxygen in the air. Cows and sheep actually need microbes to help them digest cellulose. They have those microbes in their gut. And ecosystems rely on microbes to enrich soil and actually degrade waste. So if you were... some of you have maybe heard that there are things like body farms. So people study to become things like forensic pathologists, and there are university and colleges that have body farms and actually show students different states of decomp. And it is bacteria and fungus in the soil that help with the decomp. So, and lots of people have things like compost bins. And again, it is bacteria and fungus that are actually helping do that decomposition. We actually use microbes to make tons of useful products, like wine and yogurt and cheese and bread, things like acetic acid and citric acid. Citric acid is one of the main ingredients in soda, for example, antibiotics, detergents, perfumes, all kinds of flavorings, stain removers. When you use something like a stain stick, those are actually enzymes that were isolated from microbes. There's all kinds of, take be no before and there'll be no gas later. That's actually enzymes that were isolated from fungus. So there's all kinds of cool products that are actually derived from microbes. We said in the very beginning that you had to, microbes couldn't be viewed with the naked eye. We had to actually view them with a microscope. So the very first microscope was built by an English scientist named Robert Hooke. And his microscope had about 25x for total magnification, which means he could make things 25 times bigger than they were. He certainly couldn't see microorganisms because that's not enough magnification, but he did look at all kinds of cool things. So one of the first things he looked at was he sliced a cork into really skinny pieces. And when he looked at it, he saw these little empty spaces, and he called those empty spaces cells. So because he thought they looked like these little spaces in churches in Europe where people would go to pray. So the term cell is actually comes from his work. So he published a book called Micrographia in 1665, and it contained really awesome hand drawings and really amazing descriptions, and a really great description of the microscope that he had built. But again, he never actually saw microorganisms. The first person who actually viewed microorganisms was an amazing dude called Anton van Leeuwenhoek. So van Leeuwenhoek actually lived from 1632 to 1723. Now, I would never ask you that, but first of all, I want you to know is the guy lived to be 90 years old in a time when nobody lived that long. And he actually wasn't a scientist. He was a fabric merchant. And the reason that this is important is because he's eventually going to build a microscope, but he's not building the microscope to actually do science things. He's building the microscope because he sells fabric. And at the time, people didn't go to the store and buy clothes. They would go and buy fabric, and then they would hire someone to make clothes. And he sold really great fabric, but people didn't want to pay for the great fabric. They wanted to buy fabric from someone else that was much cheaper. And he felt like if he could just show them the better quality of his fabric, they would be willing to pay for it. So he was also just a very inquisitive person, which is exactly what you have to do to be exactly what you have to have to be a great scientist. And he liked to go to the bookstore and, putter around and look at cool books. So he saw Hooke's Micrographia book, and he saw these amazing hand drawings that Hooke had done with these amazing descriptions of what he had seen, and he saw the illustration of Hooke's microscope. So Van Leeuwenhoek's idea was to build a microscope like that so he could show his clients his amazing fabric. So he built a microscope, and he was really great at grinding the lens. So to build a microscope, you have to grind the lens because that's where you get the magnification. And Hooke only got a magnification of 25 total magnification. But Van Leeuwenhoek was really great at it, and he got a magnification of 200 to 300 total magnification. This was way awesome. So he did use it to show people his fabric, and he was an amazing merchant because of it. But he also decided to do what Hooke did, which was look at all kinds of amazing stuff in his environment and start making drawings and descriptions and all that kind of stuff. So one of the first things he did was look at rainwater from a clay pot, and he wrote a description, and he said, "I see animals appearing to me 10,000 times less than those which may be perceived in the water with the naked eye." Then he scraped some plaque from his teeth. Now, we all know that even nowadays in England, people don't have the best oral hygiene. But can you imagine way back in the 1600s how bad people's teeth must have been? And he also scraped the teeth of some volunteers. And he said, "Though my teeth are kept usually very clean, nevertheless, when I view them in a magnifying glass, I find growing between them a little white matter as thick as wetted flour. I therefore took some of this flour and mixed it with pure rainwater, wherein there were no animals, and then to my great surprise perceived that the aforesaid matter contained very many small living animals, which moved themselves extravagantly. The number of these animals in the scruff of a man's teeth were so many that I believe they exceeded the number of men in a kingdom." Okay. Then I'm assuming he went to the pub to celebrate his amazing discovery. So he saw that they were changing the vat from beer, and they didn't understand fermentation at the time. Like, they made beer, but they didn't understand how it was actually done. So he looked at the bottom of the beer vat and he actually saw yeast, which we all understand now that yeast ferment and produce alcohol, but he actually saw beer. And then, much to my great happiness, because I actually studied the Giardia parasite, that summer, Van Leeuwenhoek had been very ill with explosive diarrhea with copious gas. So he decided to look at his own diarrhea under his new fancy microscope, and he said the following: I have usually of a morning a well-formed stool, but hitherto I have had sometimes a looseness of the bowels in 2, 3, or 4 weeks. So I went to stool some twice, thrice, or 4 times a day. But this summer this befell me very often, and especially when I took hot smoked beef that was a bit fat, or bacon, which food I eat with much enjoyment. My excrement being so thin, I was at divers times constrained to examine it. I see animals moving very prettily. Their bodies were somewhat longer than broad, and their belly, which was flat-like, was furnished with sundry little paws. He has perfectly described Giardia lamblia. It's teardrop shaped, and it has 8 gorgeous flagella. So he was the very first person, Anton van Leeuwenhoek, to actually see live microorganisms under the microscope. They just happened to be in his own diarrhea, which I know is a little wild, but hey, he doesn't worry about, sort of being cool. He just does his own thing. He also, by the way, looked at his own sperm and mentions that they are robust and ready to fertilize an egg. And what he decides to do, is at the time, the Royal Society of London has as its members the most famous scientists in the entire world. So he decides, and he starts to write letters to the Royal Society of London full of his sketches and observations of what he's seen. So he sends a total of 165 letters with observations and sketches. And you can imagine that the scientists there would desperately like to come and use his microscope. They would like him to teach them how to grind the glass so they can make a microscope. But he refuses, and he responds to them: my method for seeing the very smallest of animals I do not impart to others, nor how to see very many animals at one time. This I keep for myself alone. Mine, mine, mine. So he doesn't have one of the best traits of a scientist, which is, we like to collaborate and work together. So you all know that they must have just been waiting for this man to die so they could get in there and get at his lenses and his microscope and eventually try to reverse engineer this so that science could move forward, because at this point the entire field of microbiology is being held hostage by Anton van Leeuwenhoek. And then he doesn't die until he's 90. So this is unbelievably crazy. He died again in 1723, and he had magnification of about 300 total X magnification, and we didn't get 1,000X total magnification until the mid-1800s. So it took over 100 years after his death for us to reverse engineer it and move it forward. And by the way, at 1,000X total magnification, that's what we need to see the average bacteria. So we had a long time to go here to be able to actually see things like bacteria. Giardia is a parasite and much larger than something like bacteria. And I want you to think about the fact that we couldn't see bacteria underneath a microscope till the mid-1800s, and that's really going to affect how the field of microbiology is going to move forward. Because how are we going to connect all the dots if we can't see this stuff and convince people that they even exist. Speaking of which, let's talk about another interesting topic, which is spontaneous generation. So spontaneous generation is something that was a difficult topic and really went on for a really long time. So spontaneous generation is life arising from non-living matter. So according to Aristotle, it was readily observable that affidavits arise from the dew on plants, that fleas arose from putrid matter, mice arose from dirty hay, and this belief remained unchallenged for more than 2,000 years. That meat left out in the open would soon produce maggots, that mushrooms appeared from rotting wood, that rats and mice emerged from piles of litter. This is a conversation that's really important because eventually we're going to have to explain to people where microorganisms come from. So spontaneous generation is something that's going to have to be resolved for the field of microbiology to be able to be taken seriously. And life arising from non-living matter or spontaneous generation, even though that was thought to be true for 2,000 years, there were people that actually didn't think so. Very early on. So there was, in the late 1600s, there was an Italian medical doctor named Redi who did a very simple but really powerful experiment. And he wanted to challenge spontaneous generation. And he challenged the idea that maggots spontaneously arose from rotting meat. So our rotting meat would be non-living matter, and maggots would be life. So he had 3 jars. He put rotting meat in all 3 jars. And then he was going to wait to see if maggots spontaneously arose from the rotting meat. He left 1 jar completely open. He covered one with gauze. And then he sealed one completely shut. And now, if spontaneous generation is true, and life arises from non-living matter, we have non-living matter that is rotting meat, we should have maggots or life spontaneously rise from in all 3 jars. But what happened was there was maggots only in the open jar. He did publish his work, but it didn't go anywhere. And I think that part of it was because it was just meat in jars. It didn't look super scientific-y, but honestly, it was a fantastic and very well done experiment. And then, honestly, this is the late 1600s, nothing was done about spontaneous generation. People just kept believing spontaneous generation was true until Louis Pasteur came along with his experiment. It's now the mid-1800s, and Pasteur has doubts that spontaneous generation is true. He's a very famous scientist at the time. And he uses swan-necked flasks. He's a chemist, and back then, chemists a lot of times used to use glass. They learned glass blowing techniques, and he actually blew the glass and made these swan-neck flasks. So what he did is he put broth into the flasks. The broth would be something similar to like chicken broth or beef broth, and he boiled the broth long enough to kill any microorganisms in it. Okay, he boiled the bejeebers out of it and basically sterilized the broth. And then his idea was that the bend in the neck of the flask would allow air in, but prevent anything else from getting in. So, any kind of like dust or microorganisms would settle into the bend of the neck of the flask. And then at the time, Pasteur's lab was so busy, he didn't even have anywhere to keep them in the lab. So he actually put the flasks under the staircase in the building. So the staircase was open underneath, and he put them under the staircase. And every day he crawled on his hands and his knees to examine the flask, to look at the broth, to see if it was still clear or if it had gotten cloudy or turbid and microorganisms were growing in it. And he checked every day for 18 months and there was no growth. So obviously, spontaneous generation was not true because there was no growth in the flasks. So then he decided after 18 months to tip one of the flasks over and let the broth hit what had settled into the bend of the neck, and growth appeared within hours, meaning that the broth got cloudy or turbid. So he wrote a paper, Pasteur, describing his experiment, and he said in his paper, "Never will spontaneous generation recover from the mortal blow of this simple experiment." Pasteur actually always writes these sort of really, dramatic lines in his papers. So he said in his paper, life arises from pre-existing life, which was important for us to move forward in the field of microbiology. So we would say that microorganisms come from other microorganisms. Then the other things that we're going to have to do is we're going to have to link microorganisms and disease transmission, which actually people did a long time ago. So back in the 13th century, patients with infectious diseases were actually quarantined. They didn't know that it was microorganisms that was involved in the transmission, but somehow they had figured out that they were contagious and they quarantined them way back in the 13th century. In 1546, an Italian physician suggested that transmission could occur by what we now call direct human contact. He also said it could be from lifeless objects, from things like clothing, utensils, or even by the air. Again, he had no idea it was microorganisms, but that's what he said in a paper. In the mid-1700s, it was said that disease was transmitted from poisonous particles in the air called miasma. And miasma in medical school textbooks even was depicted as this like giant, weird, like skeleton and stuff like that. But we didn't know that the poisonous particles were microorganisms, but we knew it was being transmitted in the air in some way. So as the course goes on, we're certainly going to talk a lot more about disease transmission and stuff like that. But we did eventually make the link between microorganisms and disease transmission. We've all grown up knowing that diseases, like the flu and measles and chickenpox and strep throat and the common cold are caused by microorganisms. But a long time ago, people did not know that. One of the things that helped us figure that out, there's an entire field of biology known as epidemiology. There was interesting stories back or a long time ago that helped us figure this out. Was a cool Hungarian medical doctor named Ignaz Semmelweis. So Ignaz Semmelweis was a Hungarian physician, and his dream was to work at a hospital called the Lying-in Hospital in Vienna, Austria. This was the best hospital in the entire world. So to be able to go and work there amongst the best physicians in the entire world was a really amazing opportunity. So he got hired to work there, and there were two wings to the hospital. One wing of the hospital was staffed and run by the physicians and the medical students. And the other wing of the hospital was run by the midwives and the midwife students. And when Semmelweis got there, he was shocked by the number of women dying of fever following childbirth. And when he looked around, he noticed that the disease was more prevalent in the wing that was handled by medical students than in the wing handled by the midwife students. And when he actually did data, it was 29% of the women were dying of the fever in the wing handled by the medical students and the medical doctors versus only 3% handled by the midwives and the midwife students. And when he looked around, it looked, the two wings of the hospital looked identical, except for who was actually staffing the two sides of the hospital. So he thought that the mode of transmission had to somehow involve the medical doctors and the medical students. And he kind of lucked out because one of his actual colleagues died of the fever when he had been stuck by a scalpel when he was doing an autopsy. So Semmelweis reasoned that the same thing that killed his friend was on the hands of the medical students and the doctors when they did autopsies and maybe dealt with cadavers. So he was like, okay, so the source must be cadavers and the autopsies and things like that. They must be getting it and then going to tend to the patients. And the midwives and the midwife students didn't work on cadavers and autopsies. So in 1847, Semmelweis had a radical idea. He directed the medical students and the doctors to wash their hands in a chloride of a lime solution before entering the maternity ward. At this point, hand washing was not done. By the way, at this point, people do not wear gloves and they do not wash instruments at all. What they do for instruments at this point is scrape them off on the heel of their boot. So medical students and doctors wear boots and they scrape the instruments off on the heel of their boot. So this was a radical idea. Chloride of lime solution would be kind of like a diluted bleach solution. So deaths from the fever dropped by 2/3 when they did this, but the medical doctors refused to accept his new technique. And Semmelweis was forced to leave Vienna. So he actually was forced to leave the entire country, and he was beyond frustrated because, look at the data is clear. Deaths from the fever dropped by 2/3. So he returned to Hungary, and in Hungary, he actually had them use his hand washing idea again, and again, it decreased deaths from the fever, but it was still not accepted. So Semmelweis became outspoken, he became bitter, and he actually became deranged. And in 1865, he was going to be confined to the psychiatric ward in the hospital. And so he ran to the morgue, and he actually stabbed himself with a scalpel that they were doing an autopsy of a patient on. And ironically, he actually died of the fever in the psych ward. So, but he did, by the way, publish papers about his hand washing data. And we'll talk about how eventually, as you all know, hand washing does catch on. But part of it was the way that Semmelweis actually spoke to people and presented his information. So some of you might decide you really like microbiology and you're going to go into nursing, and you can be something called an infection control nurse. But part of what you have to be able to do is speak to people in a really productive way. And this was not Semmelweis's forte. By the way, I have visited Budapest, and they actually have an Ignaz Semmelweis museum, and they have all these cool things like the pitcher where he made the first chloride of lime solution, and the scalpel that he allegedly stabbed himself with, and the straitjacket that he was taken to the psych ward with, and all this crazy stuff. And I dragged my husband and made him go to it and stuff. It's awesome. If you're ever in Budapest, you have to go. I also want to talk about an emergency room physician named Jon Snow. It's not Jon Snow from the Game of Thrones, but there was someone else involved in epidemiology, but it's an emergency room physician named Jon Snow. He was working in London in an emergency room, and they were having all of these patients come in with really watery diarrhea, which, by the way, the patients actually had the infectious disease cholera. So he was calling around to other emergency rooms in London, but they weren't having the same issue. So it was really just captive to their emergency room, and he was trying to figure out what these patients had in common. So he got out a map of the local area, and he pulled all the files from the patients that had come in with the diarrhea, and he started putting pins in the map of where all the patients lived. And when he looked at it, it turned out that pretty much all of the people lived around Broad Street. And there was a couple people that lived, not right there, but when he called them, it turns out that they had visited someone who lived in that area. So when he walked around and he went to that neighborhood, people back then didn't have plumbing in their homes. They had to go down and use a pump and get water and take it up to their homes. And there was one pump that everybody was using on Broad Street. So he requested that the handle be removed from the Public Works Department because he was sure that the water coming out of that pump was actually contaminated. So as soon as they took the pump handle off and people just had to walk down about an extra block, the actual outbreak stopped right away. And then it turns out that there was inadequate sewage treatment, and there was sewage leaking into that actual water supply. So he wrote a paper, and he proposed that there was this waterborne outbreak, and that when they looked at it, it was actual inadequate sewage treatment. And he said, I don't know what the specific microorganism is, but it took another 29 years for us to identify that it was actually Vibrio cholerae that was actually causing the cholera outbreak. But what he did was epidemiology, because he actually determined the common cause of an outbreak of a disease. In this case, he did it old school. He just used a map and pins, and then they actually took off the handle and stopped the outbreak. There's an entire part of the Center for Disease Control that does the same basic things today. So both Ignaz Semmelweis and Jon Snow would be great examples of epidemiologists, and they really started the entire field for us. Vaccination, we're going to talk about much more when we get to the immune system, but I think people think that vaccination is something that's a recent thing, but we have been vaccinating for a really long time. The idea of vaccination is prevention of infectious disease. Back in the 14th century, the Chinese knew that smallpox survivors did not get an infectious disease or did not get the disease again. So what they did is they took scabs from people who had died from smallpox. They peeled the scabs off their bodies. They ground the scabs in a mortar and pestle, and they blew the powder into people's noses, and that actually worked perfectly. The Europeans knew about that practice, and they did their own twist on it. They took the dried smallpox scabs from the victims, and they placed them in tiny little slits under the skin of people. And that also worked to protect people from smallpox. So, again, people think that vaccination is a new thing, but we've been doing it for a really long time. In the 1700s, smallpox was really prevalent throughout Europe, and in England, it was killing a third of all kids before the age of 3. So, Jenner was an English medical doctor, and he worked in a really rural part of England, and he thought that a mild disease that was cowpox might provide protection against the potentially fatal smallpox. And that's because he noticed that milkmaids that milked cows would get cowpox, and they would get like blisters, just like people would get smallpox blisters, but they would get cowpox, and the cows wouldn't get sick from cowpox, but they would get them on their teats. And then a milkmaid would actually milk the cow, and they would have the cowpox on their teats. And when they would milk the teats, the cowpox would burst, and the fluid would go over the milkmaid's hands. So they would get exposed to the cowpox fluid, but he would go to villages where every single person would be dead of the smallpox except the milkmaid. The milkmaid would actually be alive, and he really thought it was because they had been exposed to the cowpox. So he actually, in 1796, again, I would never ask you these dates, he actually inoculated a boy with pus from a cowpox lesion, and then you can't just inoculate the boy with pus from a cowpox lesion and say, okay, well, that protected them, because that might not have worked. So then he inoculated the boy with pus from a smallpox lesion, and the boy was exposed to smallpox, and he did not get smallpox. And then Jenner actually repeated the experiment even with his own son. And in 1798, he published a pamphlet on this on this whole procedure, and it was very well received. It's called vaccination because of vaca, for cow, and by 1801, so just in about 3 years' time, 100,000 people in England were vaccinated this way. So again, we're going to come back to vaccination when we talk about the immune system, but I just wanted to bring it up, because this is supposed to be touching on the highlights of the kinds of things we're going to talk about, in this course. I also wanted to talk about the germ theory of disease. Again, it took us till about the mid-1800s to get microscopes with powerful enough magnifications to be able to see microorganisms like bacteria. So then the question was, could bacteria be the cause of disease? And if so, how are they actually found in healthy people? And it was actually Louis Pasteur who was the one who's going to solve this problem for us. So how this worked was France was having a big problem with wine. France had been an exporter of this amazing wine forever, but they didn't really understand how fermentation worked. They just made wine and it came out great every time, and they never had any issues. And then all of a sudden, they were having problems in getting this sour, bad-smelling wine. So they called up Pasteur, because he was the most famous scientist on the planet, and they said, come to France and figure out why our wine's not coming out good, and help us out. So what Pasteur did is he showed up in France and said, okay, I'm going to figure out what's going on. So he basically used a microscope, and he looked at wine that was good, that tasted good and didn't smell bad. And then he looked at wine that was bad, that tasted bad and smelled bad, and he saw different things. In wine that tasted good and smelled good, he only saw yeast underneath the microscope. In wine that tasted bad and smelled bad, he saw yeast and bacteria. So his hypothesis was that the bacteria wasn't supposed to be there and had contaminated the wine. So he basically said, what you guys need to do is you need to do what you have been doing, which is taking the grapes and squishing them to get grape juice. That's good. But then you need to heat the grape juice to kill any bacteria that's in it, and then add yeast back to it. So that way you don't get the bacterial contamination. Because that, as we know now, it's the yeast uses the sugar in the grape juice and ferments to produce alcohol. What they were having happen in France is they were squishing the grapes and getting grape juice, and then both yeast and bacteria were using the sugar to ferment, and the bacteria was producing acid. So in the mid 1800s, Pasteur published a paper about the wine being soured by bacteria. But in the paper, he actually implied that maybe bacteria could cause human illness as well. He basically said, hey, if bacteria can sour wine, maybe they can sour people. And it took about 5 years for Pasteur to formulate what's now called the germ theory of disease, which the germ theory of disease states that microbes are the cause of infectious disease. Pasteur eventually goes on to develop a chicken cholera vaccine, a sheep anthrax vaccine, a human rabies vaccine. He develops the Pasteur Institute in Paris, and he funds it with all the money he makes from all these great inventions. They discover the HIV virus, and they do a million other amazing things. So it's pretty, I have to say, fantastic. So now we know, for example, that the influenza virus causes the flu, and Streptococcus pyogenes bacteria causes strep throat. But to get there, basically Pasteur had to go from, and this is just how his amazing mind works, if bacteria can sour wine, maybe bacteria can sour people. But it did take him 5 years to get there. And theory is a powerful word. We don't use it very much in science. To go from a hypothesis to a theory, you have to be widely accepted by the scientific community and supported by a significant amount of data. We have the germ theory of disease and the theory of evolution in biology. That's it. So it's kind of amazing. I want to talk about Lister's antiseptic technique. So Lister was an English surgeon, and he specialized in amputation. Way back then, if you had a bad infection, we just had to cut things off of you because there was no antibiotics. There wasn't antibiotics until the 1940s. So if you had a bad infection, the only thing we could do was cut things off of you. And Lister was a great surgeon who could amputate things off of you, but half of his patients died of what we would now call a post-operative infection. And he really hated this because he hated having bad statistics. So he had read Pasteur's work on spontaneous generation. So he knew the bad stuff that was happening to his patients just didn't come from nowhere. So he began spraying his patients' wounds, the surgical incisions, and the dressings with phenol. Phenol had been used, the chemical, to reduce odor and decay in sewage and to treat parasitic infections in cattle. Phenol is really rough stuff. We're going to come back to that. It did decrease death in his patients by 2/3, so it did work. However, phenol is caustic to human skin and body tissues, and it is quite poisonous. The surgeon's skin would become bleached and numb. Their nails cracked. Their lungs would become sore because they would breathe it in in quantity. They started also having it sprayed into the air in their operating rooms. Now remember, they're not wearing gloves, so that's why their nails were, their skin was becoming bleached and numb, and their nails were cracking. And some surgeons became so ill that they stopped spraying it. Lister wrote papers, and he described it as a necessary evil incurred to attain a greater good. But he did have to stop because it was completely destroying people's respiratory tracts. And, for patients, it was like, okay, well, it's going to give you this horrific chemical burn and destroy your naked nerve endings, but you will survive. So after many experiments, he found that boric acid was a better antiseptic, but we would never use boric acid today either. But antiseptic is the idea of using a chemical on human skin that reduces the number of potentially pathogenic microorganisms. By the way, in 1928, they actually named Listerine after him, which he hated because at the time, Listerine was basically junky. Now you guys all know Listerine is basically just alcohol. But he did bring the idea of antisepsis in, and you all know we do this before surgery when you use something like alcohol or hydrogen peroxide on a wound. After, of course, you wash it with soap and water, you're doing antisepsis as well. We're going to talk about more about that later on as well. I want to talk about Robert Koch, who was a German medical doctor in the late 1800s, who went on to verify the germ theory of disease. He did a series of experiments to show that Bacillus anthracis was the cause of the disease anthrax. And his series of experiments became known as Koch's postulates. Shortly after that, 20 other pathogens were linked to their diseases between 1875 and 1900. So Koch's postulates are a set of experimental procedures that are used to prove that a specific microbe is the cause of a specific infectious disease. At the time, Pasteur's lab and Koch's lab are both working on a set of experiments to prove this microbe causes this disease. Because the idea of the germ theory of disease is out there. We're going to link this microbe to this disease. But now we have to say this is the way you're going to do it. Koch really wants to beat Pasteur at it because he just thinks like, Pasteur has gotten way too much, notoriety, and he thinks Pasteur's head is just getting too big. So Koch really wants to win. Lots and lots of sheep in Europe are dying of anthrax, or what we now know as anthrax. And when they die or they get anthrax, they can't be used for human consumption. So he decides to work on anthrax, and he comes up with this beautifully simple set of experiments that you still have to use today if you want to say this microorganism causes this disease. So here they are. The same microbe must be present in every single case of the disease. So in his case, every single sheep that died of anthrax, he could find the same bacillus bacteria in every single one. The microbe must be isolated and grown in pure culture in the lab. So they pulled the same bacillus-shaped bacteria out of every single sheep, and then they grew it in pure culture in the lab. Pure culture means a single genus and species. When we look, there's only one single genus and species, one bacteria that's growing in that. And then we have to inoculate the microbe from the pure culture into a susceptible host. So in this case, they took a perfectly healthy sheep, inoculated it with that pure culture, and the host must get the identical disease. So that poor sheep, it actually got anthrax. And then the same microbe must be re-isolated from the experimental host, and it must get the it must be re-isolated from the experimental host. It must be identical to the pure culture. So we're going to pull a sample out of that sheep, and we're going to look at it. It has to be identical to the bacteria that we gave it. If you do that, you have fulfilled Koch's postulates, and you can say this microbe causes this disease. So back when Koch did it, he said you should use the same host as what you isolated it from. So he isolated from a sheep, he used a sheep. He said if you isolated from a person, you should use a person. And back in the day, we did some shady stuff, you guys. We used people on death row in prison. We used people in the military. Now, of course, we have to get informed consent. So we didn't fulfill Koch's postulates for Giardia until the late 1970s when we actually had medication to cure them. We got people with informed consent. We put them in the hospital. We actually got diarrhea samples. We isolated Giardia from them. We put people in the hospital. We put the Giardia in water samples. We had people drink it. They got Giardia. We re-isolated, made sure it was identical to what we gave them, and then we gave them metronidazole and cured them. So we have to fulfill Koch's postulates. So that's a little bit about Koch's postulates. There are some things that you can't do Koch's postulates on. If it's something that it takes multiple microbes to do it. So something like bacterial vaginosis, which is an ecosystem imbalance, that we can't do Koch's postulates on. If it's something like AIDS, which we don't have a cure for, we can't do Koch's postulates on something like that either. Koch's lab went on to invent the Petri dish, the use of solid media, staining, photographing microbes, aseptic technique, all kinds of amazing stuff. For those of you who are going into nursing, I wanted to mention a little bit about Florence Nightingale. She lived from 1820 to 1910. I would never ask you that, but she lived to be 90 years old, again at a time when people never lived that long. She was an English nurse, and she really set the standards of hygiene and nursing care. She was the first one that really started cleaning healthcare facilities. Before that, they were filthy. They weren't clean on a regular basis. She would, her and her nurses would do things like actually sweep and mop. They replaced or cleaned the patient's clothing. They actually replaced and cleaned dressings. They actually kept statistics about giving good food. Before, if your family didn't bring you food, you just didn't get food. I mean, we all think hospital food is bad, but they didn't even feed you. She would ask her nurses to bring in a pot of food. They proved that when you fed patients, they actually got better faster. And then I wanted to mention Ehrlich's magic bullet. Once we figured out that microorganisms were the cause of infectious disease, Ehrlich was a chemist, and he was like, well, then we can come up with medications to actually kill the microorganisms and thereby treating patients. So he wanted to make these magic bullets to destroy pathogens, but remain non-toxic to humans. And the very first magic bullet he invented was to actually treat the infectious disease syphilis. And we'll talk about that more when we actually get to when we start talking about drugs and stuff like that. So that was just an overall sort of big picture situation. And a little bit of an introduction to everything that we're going to do this semester. So we'll come back to lots of those topics. But that's it for your introduction to microbiology.